Hi, I'm Blake Snyder, and I'm with the Environmental Protection Agency here in Athens, Georgia. And today I'll be talking about the influence of impervious surfaces on pathogen occurrence in human-associated Bacteroides in an urbanized stream.
Urban watersheds can often be impacted by several different anthropogenic factors. Some of these can include sanitary sewers or combined sewer overflows, which can have leaking pipes or burst and distribute fecal material directly into the streams. But we can also have impervious surfaces, and those can include parking lots, commercial or industrial buildings, or even just housing units. Um, Runoff events can often carry microbial contaminants directly from these surfaces into the receiving water body. Water quality can be negatively impacted by them. As such, fecal indicator bacteria and pathogens such as E. coli or Salmonella can be observed in very high concentrations in urban watersheds. And because of this, it has an increased public health risk for people using that body of water, whether it be through activities like swimming or fishing. The urban area that serves as our study watershed is Proctor Creek in Atlanta, Georgia. Proctor Creek's headwaters originate in downtown Atlanta. If you look over here on the right-hand side of this map, you can see downtown Atlanta, starting right in the headwaters of our watershed. There are combined sewer overflows in the watershed and also has failing infrastructure such as sewer pipe leaks. As such, it's included on the EPA 303D list for failing water quality standards. And one of these water quality standards is fecal coliforms. It's a very heavily urbanized watershed, and you can see that there is a very high percentage of impervious cover based on our sample sites here. We have several sites that are 70% or above impervious cover. We even have a couple such as Lindsay Street or North Ave, which are about 94 and 95% impervious cover. The objective of our study was to determine if impervious surface cover could be used to predict the occurrence of Salmonella, Stix2, which is a Shiga toxin that can be produced by certain types of E. coli, such as E. coli 0157H7, which can cause abdominal pain or diarrhea. We want to see if we could predict the occurrence of E. coli in terms of culturable or molecular E. coli, and also if we could predict a human-associated Bacteroides marker, HF183, which is a microbial source tracking marker that detects human fecal contamination in streams. For our methods, we collected biweekly stream grab samples from November 2015 through July 2018. We did that at 12 different sites. We analyzed for E. coli, both culturable and molecular. Molecular is qPCR. We looked for HF183, our human-associated Bacteroides, also Salmonella and Stix2. We also measured various water quality parameters such as pH, dissolved oxygen, conductivity, and temperature. Once we had collected our data, we used two different types of models to predict our dependent variables. One of them was used to predict E. coli and HF183, and that was called the Gradient Boosting Machine, or GBM model. This used many simple decision trees to ultimately determine the variables with the most influence. By doing this, we could determine which of our measured parameters are important predictors of our response variable. In this case, it would be E. coli or HF183. This was run using Virtual Beach, which was a program developed to determine relationships between water quality indicators and ambient environmental conditions. The second model we ended up using was a generalized linear model, and that was run in R. This was used to predict the expected presence or absence of our dependent variables based on predictor variables. And we used this for Salmonella and Stix2 since that data was presence-absence only. On to some results. This map shows the frequency of Salmonella in addition to the land cover classification for Proctor Creek. So as you can see, the Proctor Creek watershed is highly developed, with much of the watershed being covered in pink to dark red. and the pink to dark red symbolizes low to high intensity of developed area. The watershed has a particularly high developed and impervious cover in the upper reaches near downtown Atlanta, which is on the lower right-hand side here. But as we travel northwest through the watershed towards the pour point, you can see in the middle of the watershed that we do have some forested area interspersed throughout the impervious cover and developed area. In terms of salmonella frequency throughout the watershed, we can see that it is quite widespread throughout the entire watershed. The majority of the mainstream sites range from approximately 60 to 75% presence, and the furthest upstream site here, right here, that site had about 45% presence. But our lowest site is actually slightly downstream of it, a tributary site here, and that had only 10% salmonella presence. Moving on to Stix 2, we can see that, like salmonella, Stix 2 was widespread throughout the watershed as well. We can see the majority of the main stem sites have about 41 to 60% frequency of Stix 2. However, the highest frequency was located at the most upstream site, right here, and that was at 70% frequency, which, if you remember, that site actually had the lowest frequency of salmonella. Stix 2 was also present at the pour point of the watershed here, but that was about a 40% frequency. Shifting gears to the model results, we can see that our GBM model had very high accuracy when predicting all of our E. coli and our HF183. You can see that our accuracy ranges from 86% to 88%. Over here on the right-hand side are the important variables that were included in the model, with the percent of influence for each variable that was included. We can see that with both E. coli, impervious cover was the #1 influencer in the model, with 27 and 30% influence, followed by turbidity in both models. HF183 had impervious cover at 37% as an important variable, and followed by dissolved oxygen. So these were important variables when predicting these dependent variables. Moving on to our generalized linear model for salmonella and Stix 2, we can see that the accuracy for these models weren't as good as the ones for our GBM models for E. coli and HF183. In terms of salmonella, we had 66% accuracy when predicting the presence of salmonella. #1 in that model was temperature, followed by pH, rain intensity, and impervious cover. In terms of Stix 2, we didn't have very good accuracy predicting presence absence for Stix 2 with this model. It was 52% accuracy, and none of the variables were significant. So that model was not good in predicting the presence or absence of Stix 2. In conclusion, our study showed that impervious cover was an important predictor for E. coli, HF183, and to a lesser extent salmonella. This suggests that if impervious cover could be limited in urban watersheds, perhaps using green infrastructure practices, the presence of fecal indicator bacteria, microbial source tracking markers, and pathogens may be reduced in the receiving water bodies. Our study also showed that although Stix 2 was present in the watershed, the parameters we used failed to create a suitable predictive model. I'd like to thank Mike Satursky for his help with statistical design and Lourdes Prieto for her graphic design, and I'd like to thank you for listening. Thank you.